In the microscopic world, a silent but deadly crime unfolds every day. Normal cells, following the rules of division, suddenly go rogue. This is the case of uncontrolled mitosis.
We've got a lead. The cells in this garlic root tip are also dividing at a rapid rate. Their high meiotic rate may tell us something about this out-of-control mitosis we've encountered. Get this to forensics ASAP.
These cells are dividing fast, but what makes them different from the rapid, uncontrolled mitosis we see in our victims' cells? Let's break it down. Mitosis has 5 stages: interphase, prophase, metaphase, anaphase, and telophase. In a healthy cell, DNA is replicated in interphase, which is split into 3 parts: G1, S phase, and G2. In G1, the cell grows and carries out its typical functions. In S phase, the DNA is duplicated, and in G2 phase, the cell grows to prepare for mitosis. There are checkpoints to ensure this DNA is healthy for replication. Checkpoint number 1 occurs before S phase and ensures that all DNA is healthy and safe to replicate. Checkpoint number 2 is conducted at the end of G2 phase to make sure the cell is correctly growing, the DNA is correctly replicated, and resources are enough for the cell to begin its division. The third checkpoint occurs during metaphase to ensure all chromosomes are aligned correctly, but we will discuss that later. If the DNA is damaged, it is either repaired or it triggers apoptosis, also known as cell death. Our victims' cells have bypassed this system. There seems to be a mutation in the tumor suppressor genes, allowing the damaged DNA to continue to replicate. Therefore, these cells skip cell death even when their DNA is damaged. Now, in prophase, chromosomes are condensed, and spindle fibers begin to grow in preparation for the upcoming phases. In our victims' cells, there are chromosomal abnormalities found in prophase. In normal cells, during metaphase, the chromosomes align in the center along the metaphase plate in preparation for separation. This spindle checkpoint makes sure each chromosome is attached to its spindle fiber before separation in anaphase. In our victims' cells, the chromosomes don't align properly. Mistakes like these can lead to cells that are genetically different due to improper separation. Anaphase in a healthy cell is when the sister chromatids are pulled evenly to opposite sides. But what we see in our victims' cells is uneven chromosomal separation, resulting in abnormal daughter cells, as mentioned before. Finally, in a healthy cell, 2 identical daughter cells are formed in telophase. But as I said before, these cells we're seeing are abnormal. Our victims' cells are experiencing dysfunctional checkpoints, abnormal DNA, and abnormal daughter cells. Thanks to these rapidly dividing cells in garlic roots, we can compare their mitosis process to the abnormal cells in our victim.
I think I know who the culprit is now. Everyone, I know who committed this crime. Only one thing can cause such biological chaos: bypassing meiotic checkpoints, replicating damaged DNA, having genetically different daughter cells, and such a high meiotic index. It can only mean one thing. It was cancer.
How can we stop it?
There are many different methods, but there isn't a cure for cancer. Let's take a look at one possible treatment called Taxol. This is a drug that targets mitosis in metaphase by disrupting the microtubules, which are responsible for separating the sister chromatids. If you recall, in anaphase, the sister chromatids are separated to opposite ends of the cell. This is because the microtubules depolymerize or shorten and bring the chromatids with them. Taxol targets the microtubules in metaphase to prevent them from pulling the sister chromatids apart. Because the cell cannot divide if the microtubules do not depolymerize, it will eventually die via apoptosis. Because we understand how typical mitosis occurs due to our research with the garlic root tip cells, we are able to think of new ways to target mitosis processes in order to slow or stop tumors' growth.
And so, the verdict is in. The rogue cells, guilty of uncontrolled division, which ultimately led to cancer in the victim, have been sentenced to Taxol, ensuring they meet their rightful end. Justice in the microscopic world is swift and final. I would like to say a special thank you to Professor Joo-Hyun Lee and Professor Soo-Hwa Lee. Without your guidance, this case would have never been solved.